Okay, as a quick revision before I start concentrating on compound microscope, now you know the definition of a microscope. We know that there are 3 types of a microscope and we know the differences between them. But now when it comes to learn about the body parts of a microscope, I need you to concentrate on compound microscope. This is the one that matters for me because this is the one that we are using so far. Now, in order to understand the body parts of a microscope, I need you to go from the bottom over here. This is what we call it as base, the base of the microscope, okay, where it carries all the body parts of a microscope. This is the light source over here, okay, and if you can see, this knob controls the intensity. I need just to show you. This is the intensity of the light itself. Now under the microscope over here there is a lens. I cannot show it to you, I cannot remove it, unfortunately. Usually when you are in the lab you can just bend over and look at the lens. This is a reflect lens. It reflects the light over here to the condenser lens, and from the name it is it condenses the light with the lever over here. See how it condenses the light? This is where we apply the slide, okay? This is, imagine this, my hand is the microscopic slide, and the lens over here will magnify the image in the slide, and through these lenses you can view the image. So basically, as I said, it's light and it's lenses, reflect and zooming lenses, okay? So before we know how to use the microscope, I need you to understand the basic building parts of a microscope. This is the base, this is the light source, this is the stage, and I control the movement of the stage by mechanical knobs. In your assignments that I will give you on or in the picture, you have A and B. This is just right and left, so both of them you can call them as mechanical knobs, okay? I have here 2 knobs, the big one and the small one. This is the big knob. Look at the big knob, it controls also the stage. It takes it upwards and downwards, okay? And this is the sharp or fine focusing, so coarse, fine focusing knob. This is the arm of the microscope that carries the stage. And over here I have the black round, you cannot see it. This is revolving nose piece. It carries the objective, and from the name it revolves in order to move from one zoom to another. These are the objectives, lenses, and the lenses are magnifying lenses attached to the revolving nose piece. Now this is the head of the microscope that carries the oculars, and to define oculars, these are lenses that you look through. So this is better, okay? From top, oculars, lenses that you look through. Head, it carries the oculars. Revolving nose piece, it carries the objectives. What are the objectives? Are magnifying lenses. This is the arm of the microscope that carries the stage. This is the lever where you hold the slide. The stage carries the slide, okay? This is the mechanical knob. A and B both are called mechanical knob in order to move the stage. Under the stage, right away, this is a lens. It's a condenser lens. It condenses the light. Can you see it? This one. Now, this lens over here, which I cannot remove, it's called iris diaphragm. This is the light source. This is the base of the microscope. And I have two knobs. Those knobs are for the light, basically. This is the light intensity. And in the back, I have the light switch. Now, sometimes you find both of them in the back, both of them on the base. This is depend on the manufacturer itself, okay? Now, for the procedure, how to use the microscope, it is very simple. This is the microscopic slide. This is fixed, this is permanent, and this is where we have over here the sample itself under the cover slip. All what I have to do is that you have to sit and face the microscope. Very simple. You will apply the slide on the stage and you will hold it. You don't put the lever of the clip. This is the clip. You don't put it on top of the slide. You will break my slides. So you will just simply do this way. Now it is fixed on the stage. With the help of the mechanical knobs over here, I will move the slide in a way that, now you cannot see it over here, but from my side, I am actually facing the cover slip over here in between the condenser and the objective lens. Once I reach this way, I will put my eyes through the oculars and use the coarse focusing to pull the stage toward the objective. Now, why I choose the smallest objective over here? Because no matter how I keep, pulling the coarse focusing stage, it won't touch the objective. I always start with a lower magnification, 4x, because if I use 10x while I'm pulling the stage with the coarse focusing knob, and I have to keep my eyes through the oculars to see the image, you need to be careful that you won't touch the slide and you won't break the slide, okay? So to be on the safe side, we use the lower magnification and your eyes through the oculars. Once you see the image, you will stop, even if it's an unclear, and you will sharpen the image by using the fine focus. And from there, you can look at the image, you can draw it, and then if you need to go for a higher magnification, very simple. All what you have to do is to pull the revolving nose piece and change it into a 10, into a 40, very freely. You can remove it. All what you have to do is to sharpen the image by using the fine focus, the small knob over here. Because once you go deeper, it becomes really sometimes foggy, so you will just have to focus it. But when you will use the 100, I have to use a certain oil, just one drop on top of the slide, because the 100, objective 100, will touch the slide. And that will cause a friction and we will break the slide by then. So the oil will facilitate the movement of objective 100 only while using objective 100, okay? But for 4x, 10x, 40x, I can move the revolving nose piece freely and without even touching the slide. But for 100, it will touch it. Now, because I didn't fix the slide itself, but I will show you now my point. Now I have fixed the image and I can see it under objective 4x. Now I need you to look, I will show it to you. Now when I move it, see, still it won't touch. It's not touching. But when I go for objective 100, it will touch the slide. See, I won't go furthermore. I need to add a drop of oil. It is called oil immersion for avoiding breaking the slide because it will cause a friction. So the oil immersion is to facilitate the movement of objective 100. That's it. Now I know it is hard for you to understand without looking at the microscope and working by your own hands. I hope I actually clarify how to use the objectives and the body parts of microscope. That's all. You have a very small assignment to solve and send it to me. That's all for today, dear. Now for cheek cell, same 2 drops, water and stain, but in this case I need to do onion. Did I say cheek cell at the beginning? Yes, onion, real life onion. Now, why did I cut the onion? Because when I cut it, I need to peel this peeling over here, the outer peel. Can you see it? Very thin, or from inside. And this is better, much more easier. Look, this is the one I'm using. See? This is the one I'm using. So I will just apply it on the water. And I have another piece over here. See? This one. I will apply it on and I will add my cover slip as well. Again, I will tilt and try not to drop, very slowly. See? Nothing. Very slowly. I will try to tap to remove this air bubble, and then it is ready to use under the microscope. See, I will just tap it. No air bubble. And this is cheek cells and onion cells ready to view under the microscope. Now, to prepare fungus sample, this is rhizobus. It is a black mold fungus, bread black mold. Remember, if you ever actually experienced black mold on top of the bread, we removed that bread and we add it in conservative. This is alcohol. Now, why do we add it in alcohol? We call it fixation in order to make the sample harmless. So this is why we add it in alcohol, let's say. It's a fixation. Fixation, we're adding the sample in alcohol to make the sample or the specimen harmless in order to work with. So since this is liquid, I either add directly from here into this slide and add stain, or you can just use it without a stain. It's very simple and very clear as well. But I will do both samples. So again, no need to put a drop of water. I will add very small drop of stain because it's going to be a bit messy if I add too much liquid. I will try to mix what's inside the vial in order to have the sample, and I will take very small drop over here, and I will drop one drop over here with the stain. I'm I'm gonna give it a little bit mix, and I will add my cover slip, but this is large. Oops, it's very large. You need to wear your size, but this is the one I can find right now. I'm careful, of course, because this is real fungus that I'm using. See, that's it. This is a fungus sample ready to view under the microscope. Very simple wet mount slide preparation, very simple experiment. It's a slide making to view cells under the microscope, and it's temporary slide. If I need to study that slide later on, all what I have to do is I will add glue to adhere the cover slip on top of it, and then I will call it a permanent slide. That's it. Okay.